Welcome to the deep dive. Our mission here is pretty simple. We take complex source material, cut through the jargon, and pull out the key nuggets of knowledge you need, nice and quick. Today, we're diving into agricultural ecology. Specifically, we're looking at a really interesting research paper led by Naoki Katayama. It's titled Complementary Role of Environmental DNA for Line-Transect Bird Surveys, a field test in a Japanese rice landscape. Bit of a mouthful.
It is, but that title really lays out the mission, doesn't it? The core idea is comparing this newer technique, environmental DNA or eDNA metabarcoding, with the classic method, walking a line and counting birds, the line-transect survey. So yeah, our deep dive is all about figuring out how useful this eDNA approach is. Specifically, when you get the DNA from water in rice paddies, can it actually give us the data we need on bird communities?
And the focus on rice fields, that's not just some random choice, is it? These paddies are huge globally.
Oh, absolutely not random. Rice fields cover something like, over 20% of the world's cereal area. They're massive ecosystems.
And they act like artificial wetlands, providing vital services, pest control, nutrient cycling, that sort of thing. Plus, they support a ton of wildlife, especially birds.
Exactly. And that context is critical because we're seeing serious biodiversity loss in these agricultural landscapes. Think intensification, climate change. It's hitting these ecosystems hard. And birds are a really key group here. They're sensitive indicators, and many are threatened. So getting good data is, essential if we want sustainable farming.
Okay, so we need good data. Why not just stick with the traditional methods? Having an expert walk a path, listen, watch, seems pretty thorough.
It is thorough in some ways, the gold standard even, but it's got some pretty significant limitations built right in. First off, it's super labor-intensive, takes a lot of time, a lot of effort.
Right, and you need real experts, people who can tell maybe very similar looking or sounding species apart instantly.
That's a huge bottleneck. But maybe the biggest limitation, and this is what the study really gets at, is what these surveys miss.
What do they miss?
Well, they usually happen during the day, right? So they're often not great at detecting birds that are rare or really well-hidden cryptic species or, crucially, nocturnal ones.
Ah, the night shift. The ones that only come out after the surveyors have gone home.
Precisely. Think about something like the black crown night heron. We often don't have great data on them because, they're not active when people are out surveying at 10 a.m.
Okay, let's unpack this. So there's a real gap, a need for methods that maybe aren't so reliant on expert eyes and ears and can capture more of the full picture.
Exactly. There's a need for something maybe easier to implement, potentially cheaper, and definitely something that can catch those elusive species.
And this is where environmental DNA, eDNA, comes onto the scene. It's relatively new, isn't it?
Relatively, yeah. The idea with eDNA metabarcoding is you collect environmental samples, in this case, water, and you look for tiny traces of DNA shed by organisms. Skin cells, feces, feathers, you name it. In these flooded rice fields, the paddy water itself is like a soup collecting all this DNA. So the potential is there to detect species without ever actually seeing or hearing them.
Okay, so let's get into the study itself. How did they set it up? It was in central Japan, right?
Yep, Kaminakawa Town, Tochigi Prefecture. They did the work during the bird breeding seasons in 2021 and 2022. And the landscape sounds pretty typical, a mix of rice fields, other farmlands, some grassland, irrigation channels.
A mosaic, basically.
A classic agricultural mosaic. And they ran the two methods head-to-head.
So method one, the traditional approach.
Right, the line-transect survey. An experienced observer walked the same 1.5-kilometer route repeatedly during the day, recording all the birds they saw or heard, species, number, location. And they used that data to estimate bird biomass using distance sampling.
Okay, distance sampling. That's where you estimate population size based on how far away you detect the animal, yeah. More distant detections suggest lower density, closer ones higher density.
That's the core idea. It gives you a quantitative measure of abundance from the visual survey, so that's the baseline.
And native in two?
Yeah.
The eDNA.
For the eDNA, they collected water samples, took them from 11 different rice fields, plus the irrigation channel running alongside them, multiple times through the season. They took these samples, filtered them up to 500 milliliters of water per sample to capture the DNA fragments.
And then in the lab, they looked for bird DNA specifically.
Exactly. They used a standard set of genetic primers called MIBIRD, which targets a piece of the mitochondrial 12S rRNA gene. It's a region that's pretty universal in birds, but varies enough between species to tell them apart, like a barcode.
Got it. So they get lists of species from both methods. What were they comparing statistically?
Two main things. First, just the raw number of species detected by each method, the species richness. Second, They looked at how often a species' DNA was found across the samples, the occurrence frequency, and tried to link that to ecological traits.
Like what traits?
Specifically, the estimated bird biomass from the transect data and the bird's general habitat preference. Was it mostly an aquatic bird or a terrestrial one?
Okay, moment of truth, the results. How did they stack up just purely on species count?
Well, if you just look at the total number of species detected, the traditional line-transect survey was clearly ahead. No contest, really.
How far ahead?
The line-transect picked up 18 species in total. That broke down into 12 terrestrial species and 6 aquatic ones.
And the eDNA?
The eDNA from the paddy water, only 8 species total, 3 terrestrial, 5 aquatic.
Wow, okay, that's less than half.
Yeah. And here's the kicker. 12 species were only found by the line-transect survey. They just didn't show up in the water DNA at all.
That sounds pretty damning for eDNA, doesn't it? If it's missing more than half the species richness, is it really that useful on its own for a full biodiversity assessment?
That's a critical point. The study makes it clear, eDNA from paddy water alone is not a replacement for traditional surveys if your goal is capturing the entire bird community.
But I sense a but coming.
There's definitely a but. This is where the complementary role part of the title becomes key. Because the eDNA didn't just find fewer species, it found some different species. Here's where it gets really interesting. 2 aquatic bird species were detected only by the eDNA method. They were completely missed by the expert observer doing the daytime transects.
And which species were those?
The black-crowned night heron and cormorants, likely the great cormorant, common in the area.
The night heron. That's the nocturnal one you mentioned earlier.
Exactly. That finding is huge. It's the proof of concept. It shows eDNA succeeding precisely where the traditional method failed, detecting a species active outside the normal survey hours.
So it fills a specific known gap.
It demonstrates that potential perfectly. The cormorant is interesting too, possibly linking the paddy fields to nearby rivers where they might be more common. So the picture isn't just fewer species, it's a different slice of the community. Combine the methods, and you get a more complete view.
Okay, that makes sense. It's not better or worse necessarily, but complementary. So why the difference? Why does eDNA pick up the heron but miss, say, a sparrow that the observer saw? Did the stats give any clues?
They did. The analysis pointed to 2 main drivers, biomass and habitat.
Let's take biomass first. How did that relate?
There was a clear positive link. The frequency of DNA detection, how many samples a species showed up in, was significantly associated with its estimated biomass.
So bigger birds equals more detectable DNA.
Essentially, yes. Larger species, maybe like crows or doves, which have higher biomass, tended to be detected more often by eDNA.
And the little guys?
The small birds, sparrows, swallows, wagtails, the ones with low individual biomass, they were often missed completely by the eDNA, even if they were seen on the transects. They just don't seem to shed enough detectable DNA into the water during brief visits.
Okay, that tracks. What about the habitat link? Aquatic versus terrestrial.
That was even stronger, really. The occurrence frequency in the water samples was significantly higher for aquatic species compared to terrestrial ones.
Which makes intuitive sense, right, if you're sampling water.
Exactly. Aquatic birds, things like ducks, egrets, herons, they spend a lot of time in the water. They're foraging, breeding, resting, defecating directly into the medium being sampled.
So they're constantly depositing DNA.
Much more intensively than a terrestrial bird that might just perch on a levee or fly over or take a quick drink. The contact time and intensity seem crucial for DNA deposition and accumulation in the water.
Okay, so eDNA is better for bigger birds and those that spend a lot of time in the water. What about abundance? In some fish studies, the number of DNA sequence reads relates to how many fish there are. Did that work here?
Not really, no. That was another interesting finding. The total number of reads for a species didn't clearly correlate with the estimated biomass from the transects. So you couldn't reliably say more reads equals more birds.
So not great for counting individuals.
Not for direct counting, it seems. But the researchers suggested an alternative idea. They saw really high read counts for species like the eastern spot-billed duck and the intermediate egret.
Both very active in paddies.
Exactly. So maybe, they proposed, high read counts aren't indicating numbers of birds, but rather high levels of activity by those species within the sampled fields. Which could still be ecologically meaningful, you know.
Yeah, like indicating which species are most heavily using that specific habitat patch, perhaps for foraging. That's useful info.
Definitely. Now, we should also touch on the study's limitations. The researchers were upfront about them.
Like the fact that eDNA just didn't find as many species?
Right. They did some extrapolation like, what if we doubled the number of eDNA samples? And even then, the predicted species count, maybe 11 species, was still well below what the Transect survey actually observed, like 17 species in 2021.
So even with more sampling, the eDNA method, at least from paddy water, seems unlikely to capture the full richness found by the transect.
It seems that way, yes. And another methodological point. The transect route covered a bit more ground, including adjacent habitats like grasslands and maybe some woodland edges.
Which would naturally favor the detection of terrestrial birds that might not venture into the flooded paddies themselves.
Correct. So the comparison isn't perfectly apples to apples in terms of the exact area surveyed, which could explain some of the discrepancy, especially for those land birds.
Okay, so pulling it all together, what are the big implications here for conservation, for monitoring?
The main conclusion is pretty clear. Conventional surveys, like line transects, are still probably the most rapid and comprehensive way to get a snapshot of overall bird diversity, especially for terrestrial species. They give you that broad overview.
But they have blind spots.
Big blind spots. And that's where eDNA comes in as this vital complementary tool. It's particularly powerful for those aquatic species, and critically for nocturnal or cryptic species that the standard surveys consistently miss. And remember that finding about the irrigation channel. Water samples from the channel often had higher detection rates and picked up species, like the cormorants, potentially linked to nearby rivers.
Right, so sampling strategy matters.
Hugely. It suggests that for a really good landscape scale assessment, especially if you're interested in aquatic birds, you shouldn't just sample the paddy fields. You need to sample the connecting waterways to the channels, maybe nearby ponds or rivers.
That broader aquatic network sampling seems key if you want to evaluate things like how organic farming versus conventional farming impacts the whole bird community, not just the ones right in the field.
Precisely. That landscape context is crucial.
So what does this all mean? What's the final word?
It means we have a clearer picture of the trade-offs. We know conventional surveys are good for breadth, especially on land. We know eDNA from water is fantastic for depth in aquatic systems and for catching those hard-to-see species. Better bird biodiversity information, using both methods smartly, will serve as a basis for improving the sustainability of agriculture. It gives us the tools to really understand what's going on.
So the study shows eDNA from paddy water isn't a silver bullet replacement, but it's an indispensable partner for conventional methods, especially for water-loving and nocturnal birds. But thinking ahead, science doesn't stand still. We're hearing about eDNA being detected from the air now, even collected from flowers visited by insects or birds.
Yeah, the field is moving incredibly fast. Air DNA is a fascinating frontier.
So here's a final thought for you to ponder.
Yeah.
If that air-based eDNA technology matures, could we reach a point where we can get a rapid, comprehensive snapshot of the entire bird community, terrestrial species, tiny birds, everything, just by sampling the air, maybe even surpassing what line transects can do today?
That's the million-dollar question, isn't it? It's certainly an exciting possibility for the future of biodiversity monitoring.
Something to keep an eye on. Thanks for joining us for this deep dive.